One of the fundamental characteristics of life is reproduction. Life forms on Earth reproduce in different reproductive mechanisms, but all of them maintain similarity by being able to copy their genetic material as the basis for biological inheritance. This process is known as DNA replication. In 1953, Watson and Crick, absolutely with Franklin and Wilkins' contribution, discovered the structure of DNA as a double helix, as well as the different novel features of this so-called blueprint of life. Importantly, the two scientists hypothesized that the double helix unwinds, and each of the parental strands serves as a template for the synthesis of a new complementary strand. This enables the mechanism for replication according to them. After Francis and Crick's discovery and assumption, two other models for DNA replication have been proposed by the scientific community at that time. We have the conservative and the dispersive model. Conservative, one daughter molecule contains both parental DNA strands, and the other daughter molecule contains both newly synthesized strands. For the dispersive, parental strand is broken into fragments, resulting to parental and progeny DNA segments interspersed. And for semi-conservative, each daughter duplex contains one strand from the parental structure. To know which of these three would be the accurate model, in 1957, Mendelssohn and Stahl conducted an experiment. They cultured bacteria in a heavy isotope of nitrogen, thus yielding dense DNA. After that, they shift the culture to a light isotope of nitrogen medium. After the first replication cycle using cesium chloride, equilibrium density gradient centrifugation, they saw that the DNA yielded intermediate density, thus eliminating the conservative model. After another round of replication cycle, the DNA yield lighter density replicates, but the intermediate density still remained, thus eliminating the dispersive model, concluding that DNA synthesis is semi-conservative. Moving on to bacterial replication, the process starts after the necessary proteins bind at the origin. Then the replication fork will start to move bidirectionally. The requirements for the overall process are primer strand with 3' hydroxyl ends, which carries out a nucleophilic attack on the incoming nucleoside triphosphate. DNTP substrates, which eventually forms the base pairs adenine, thymine, guanine, and cytosine. The enzymes, which we collectively call as replisome, which includes helicase, SSP, primase, and the DNA polymerase. There are three major stages for DNA replication. We have initiation, elongation, and termination. Initiation begins at ORIC, which is the origin of E. coli. DNA A protein recognizes ORI sequences and opens the duplex, which allows the attachment of helicase to unwind the replication bubble. As the DNA unwinding occurs, torsional strain is generated but is relieved by the DNA gyrase. Overall, there are two replication forks moving away from each other. Next, to elongate, there are two necessary requirements. There should be an RNA primer strand to which nucleotides can be added and a template DNA strand to copy. The primer is added by enzyme primase, which has 3' hydroxyl group where DNA polymerase 3 can start adding DNA nucleotides. DNA polymerase 3 is the major replication machine which have its beta clamps to keep it associated with the DNA template. If the synthesis occurs only at 5' to 3' direction, how does the lagging strand being synthesized? The answer is through Okazaki fragments, which are short DNA sequences synthesized discontinuously. The RNA primers are subsequently removed by nuclease activity of DNA polymerase 1, and the resulting gap in the strand is filled with DNA by it also. And then, it is sealed by the DNA ligase. Overall, the replication is semi-discontinuous. Lastly, termination occurs when replication fork stops at the terminus region. Then, topoisomerase 4 cuts or decatenates the link between two circular DNA. Next, the viral replication of SARS-CoV-2, which is the causative agent of the COVID-19. There are 5 major steps for viral replication. First is the attachment. SARS-CoV-2 attaches its S protein to complementary receptor sites on the host cell surface, which are the angiotensin-converting enzyme 2, or the ACE2 receptors found in the epithelial cells of lungs. Next is the entry. It enters via receptor-mediated endocytosis, wherein the virus is incorporated through the continuous invagination of the plasma membrane, forming a vesicle and closing the virus. For uncoating, the viral nucleic acid separates forming its protein coat via the action of lysosomal enzymes. The major and important process, the biosynthesis and maturation, its single-stranded RNA acts as mRNA. Because it is a positive sense strand, it can be immediately translated via the use of host cells' ribosomes. The single-stranded viral RNA translates two major proteins. The first is the one that stops the host cell's synthesis of RNA, and the second is the RNA-dependent RNA polymerase, which complements the sense strand to produce antisense strand, which will be used as a template to produce more copies of the viral RNA. Via this continuous transcription of the antisense strand, we can yield subgenomic sense strand, which will translate into viral proteins using the endoplasmic reticulum machinery. When viral proteins are produced, we can incorporate the genetic material of the virus and modify them using the Golgi complex machinery. Thus, maturation occurs, forming the viral progeny. Lastly, for release, the release of the viral progeny involves the secretory pathway. From ER to Golgi complex, the assembled viral progeny will bud out into a vesicle. The vesicle then will be subject to exocytosis in the plasma membrane.
We now move on to eukaryotic cells. So generally, both prokaryotes and eukaryotes follow the same steps for replication. But there are distinctions because eukaryotes have larger and complex linear DNA as compared to the circular DNA of prokaryotes. One major difference is that eukaryotic cells have many origins of replication. Therefore, initiation in the cell occurs in many sites. To allow this difference, eukaryotes have unique portions in their genome called replicons. Each replicon has its own origin from which replication forks proceed. The existence of replicons was first demonstrated in an autoradiographic experiment. This is shown in the image, in which the DNA is replicated simultaneously at several sites. The lines indicate sites with incorporated radioactive DNA. Initiation begins in the center, forming two replication forks that travel away from each other until they meet a neighboring fork. There are three general characteristics of DNA replication in eukaryotes. First, initiation of DNA synthesis is subject to regulation to prevent the reinitiation of a site that is already duplicated. Second, activities that occur in replication forks are similar in all cells. And third, there are 5 DNA polymerases isolated from eukaryotic cells. They are designated as alpha, beta, gamma, delta, and epsilon. The underlined polymerases are involved in nuclear replication, while polymerase beta functions in DNA repair and polymerase gamma replicates mitochondrial DNA. In this table, the proteins involved in replication are shown. Both replication systems require some similar enzymes and varies in some, but these still perform similar functions. It is better to pause and read the table first before proceeding. Polymerase delta is the primary synthesizing enzyme of the lagging strand, while it is the polymerase epsilon for the leading strand. The pCNA is a sliding clump for both polymerases. It is loaded on the DNA by the protein RFC. On the other hand, RPA is a DNA binding protein comparable in function to that of SSP in E. coli. Then the RNA DNA primers of the lagging strand synthesized by the polymerase alpha primase complex are removed by the movement of polymerase delta, generating an RNA DNA flap that is removed by the FEN1 and the nuclease. The gap is then sealed by the DNA ligase, and the topoisomerase remove the positive supercoils that develop ahead of replication fork. Like bacterial polymerases, All of the eukaryotic polymerases elongate DNA strands in the 5' to 3' direction. To ensure the accuracy of replication, polymerase gamma, delta, and epsilon possess a 3' to 5' exonuclease. The chromosome can be broken down into chromatin, which is composed of nucleosome made out of protein called histones. Examination of replicating DNA reveals nucleosomes on both daughter strand duplexes very near the replication fork, indicating that the reassembly of nucleosomes is a very rapid event. DNA is very susceptible to environmental damage. Therefore, different repair mechanisms are known. One of these is nucleotide excision repair. In this repair system, damaged bases are cut out and replaced with DNA as directed by the undamaged template strand. This is used to remove pyrimidine dimers and chemicals such as those gained from smoking. Read the steps for further details. In base excision repair, only the damaged base is removed. It is initiated by DNA glycosylase that recognizes alteration. DNA glycosylase removes only the base and not the entire nucleotide. Although many errors are corrected by polymerase proofreading, mistakes in DNA replication can still happen. Thus, any remaining mispaired bases are replaced by mismatch repair. A protein binds to the mispaired base and a second complex coats the DNA. DNA polymerase then replaces the missing section with correct nucleotides, and the DNA ligase seals the gap. Some factors can cause the splitting of the double-stranded DNA into two. Two pathways involved in the repair of the TSP breaks are the non-homologous end joining and homologous recombination pathways. In non-homologous end joining, the two broken ends of the chromosome are simply glued back together. This is messy and typically involves the loss or addition of a few nucleotides at the cut site, so this tends to produce imitation. In homologous recombination, information from the homologous chromosome that matches the damaged one is used to repair the break, as seen in the picture.